The thing that gets me out of bed in the morning is not thinking the more I work on aging, the better my own chances of making the cut and, benefiting from the achievement of longevity escape velocity. Because I recognize that even though I am doing quite a lot to hasten the defeat of aging, nevertheless, in numerical terms, the amount by which I am improving my own chances is pretty modest. A few percent if I'm even quite optimistic. It's a few years I might bring forward the defeat of aging, but, I might already be young enough that I'm going to make it anyway, even if I were doing something else with my life. Or conversely, I might be too old so that however fast it happens, it won't be in time for me. So that's a difficult thing to get excited about. Whereas, if I look at the whole world, 110,000 people die of aging every single day. So every day that I bring forward the defeat of aging, I'm saving 30 World Trade Centers, and that's quite easy to get out of bed for.
Welcome to Galaxy Balance, a podcast exploring the science of living systems, the future of intelligence, and how we maintain perspective while working at the edges of human possibility. I'm your host, Corey Smith. Today's conversation tackles one of the most profound questions we can ask. Is aging inevitable, or is it an engineering problem waiting to be solved? Today I'm joined by Aubrey De Grey, one of the most influential and controversial thinkers in the field of aging and longevity. Aubrey is a biomedical gerontologist and longtime architect of the SENS framework, Strategies for Engineered Negligible Senescence, which reframes aging not as a mysterious process, but as an accumulation of specific, repairable forms of biological damage. Trained originally as a computer scientist, Aubrey brought an engineering mindset into biology at a time when the idea of treating aging itself was widely dismissed. Over the past 2 decades, his work has helped shift that conversation from the fringes of science into a serious academic, biotech, and investment circles. In this episode, we'll explore aging as a systems problem, the role of stem cells in rejuvenation, and how we should measure rejuvenation, the impact of AI on longevity research, and what a world with radical, extended, healthy lifespans might look like. Aubrey, welcome to Galaxy Balance.
Well, thank you for having me, and that's a lovely introduction. I'm delighted to be here.
Excellent. Happy to have you on the show. So I want to start with a little bit more about your story and your arc. So if you could walk us through what originally convinced you that aging and treating it as an engineering problem was something you wanted to dedicate your life to.
Really, that happened when I was in my late 20s, prior to which, as you say, I was a computer scientist. And the thing that changed when I was in my late 20s was that I had recently married a biologist who was a lot older than me. She was already a full professor at UC San Diego. She was in the UK on sabbatical. And through her, I learned a lot of biology by accident over the dinner table. But I gradually began to realize that we were never talking about aging. And so I would ask her about that, and I would say things like, you don't seem to be interested in aging, and she would say, no, I'm not. And I would say, well, wait, why not? And she would say, well, it's just decay, isn't it? What fundamental truths about the universe are you going to, discover by studying decay? And I would say, well, sure, but it's bad for you. And she would say, that's not my problem. And I would say, well, it kind of is. And that would be as far as we would get. And this was an enormous shock to me, because the reason I had not worked on aging all my life was simply because I didn't consider myself to be a particularly talented biologist, whereas I did consider myself to be a pretty good programmer. And so I thought, play to one's strengths. There's going to be people who are good biologists who are going to be working on aging, because it's obvious that aging is the world's most important problem. And it was only during these kinds of conversations that I began to realize that the idea that aging was the world's most important problem was not universally shared. I've progressively discovered that most biologists thought the same way that my wife did. And I thought that won't do. So eventually, after a couple of years of coming to terms with that whole phenomenon that I found a complete bombshell, I thought, well, I've got no choice really, I've got to switch fields. And I knew that there's a nice distinguished history of people switching fields and doing important stuff in their new field, because they're not encumbered by the conventional wisdom and so on. And I knew that I was pretty good at working on hard problems, because my work in artificial intelligence research had gone quite well. So I thought, I've got a chance of making a contribution. So I thought, I'll have a go. And here I am.
Excellent story and how you got there. And as a computer scientist, what do you think are some of the challenges with longevity that a lot of biologists might overlook? As you say, coming into it from a different angle, you have a different perspective that might be blind alleys for a lot of classical aging researchers.
So for me, it wasn't exactly because I was a computer scientist. It was because I was an engineer. Before I started having these conversations with my wife, I did not really grasp the absolutely profound disparity of mindset that exists between technologists, shall we say, and basic scientists. That, technologists are all about humanitarian benefit. They're all about figuring out how to manipulate nature. And, of course, they understand that you need to understand a certain amount about the system you're trying to manipulate before you have a chance of being able to manipulate it rationally, but only a certain amount. There's a point where you know enough, or at least you might know enough. You think you might know enough, and therefore you'll have a go. Whereas in basic science, it's just not like that, because people find things out for the sake of finding things out. And so that means that, the best questions are the ones whose answers create nice new questions, and the idea of knowing enough just doesn't compute. So that was the most fundamental thing that guided me. And certainly when I started getting involved in the field of the biology of aging, I found that nearly everyone in it was basically going about it as a basic scientist. They'd essentially given up on the idea of ever being able to do anything about aging. It was, when I'm in my more frivolous moments, I tend to compare gerontologists of that era to seismologists. People who work on earthquakes, they know that earthquakes are bad for you, but they have no aspiration at all to, stopping them from happening. So, that's changed now, of course, quite a lot, but it was very much the mindset. And so, yes, I was able to come in and look at things in a different way, and it didn't take me all that long to see a variety of things that other people were missing.
Yeah, I love your analogy about the seismologist that can't do anything about it. I think the times are changing with many companies coming up and actual approaches and therapies coming along to address this. Is it just technological innovation that enables it, that people can see that it's closer, you think? Or what enabled the change in mindset since you started working on this problem?
Yeah, I think it's all of the above, really. I think the progress in the laboratory and in the clinic has definitely helped. Every small step forward makes it easier to believe, so to speak, that we are within striking distance of the whole thing coming to fruition. It's also that experts are speaking about it in a different way. When I started out, I was very much a voice in the wilderness. Certainly in academia, people thought that I was, first of all, it was a case of having to justify that what I was saying was even scientific. But even once I had essentially won that argument, it was still quite difficult to get most of my academic colleagues to, they would tend to want to keep their distance from the whole idea. Just because they thought it would be, harmful to, it would be politically incorrect, harmful to their funding, things like that. But yes, things have dramatically changed. It's become, in fact, in one sense, in academia, it's gone complete 180. That, you could not mention intervention at all in a grant application 20 years ago and have any chance of having it funded. Whereas now, it's the other way around. You kind of you're obliged to talk about the translational potential of what you're trying to do. And in the wider world, this has all happened hand in hand with the wider world. So of course, I've been out there in the mainstream media and so on, and doing just a completely amazing number of talks and podcasts and so on since about 20 odd years ago, and it has added up. Just the repeat advertising does help, and of course, there's more and more people out there now saying the same kind of thing, and often saying the same kind of thing in a different style than I say it, which is great because of course it reaches, it resonates with different audiences and so on. And as time's gone on, the private sector has become more and more involved. Of course, that's definitely made a huge difference. I think your former mentor George Church has played an enormously important role in this by encouraging and leveraging his own academic position to allow people from his flock, including, of course, you, to go out and start companies. I think it's well over a dozen now that have mostly done pretty well. And so, the more of that, the better.
Excellent. I want to dig into this idea of aging isn't the greatest problem. You mentioned that getting into classical biologists, they didn't agree with that. And from an evolutionary perspective, I kind of agree with them that if organisms live long enough, there's no opportunity to evolve and acquire mutations and select. But from our human and as society, I definitely acknowledge the grand challenge of approaching death. Spirituality, all of it's built around accepting that in many cases. But like you, I agree that this is a problem that should be tackled and should be addressed. And what do you think society may be like if we implement radical life extension?
Yeah, so I don't really think all that much about what society is going to be like in a post-aging world. And the reason I don't is because it is going to be quite a long way away. We are looking at maybe 12 to 15 years before we have even as much as a 50-50 chance of getting to this threshold that I've called longevity escape velocity. And after that, even if we have almost nobody dying because aging is not happening anymore, then, still people are only going to be getting older at 1 year per year, so things will change fast. It'll change slowly, is what I'm saying. And a lot of other things happen during periods that are that long. I think it's basically pointless to think about the distant future in terms of how things are going to be. All I think we can really rely upon is the fact that nobody is ever going to want to get sick, however long ago they were born. And if you don't want to get sick, then and we have the technology to stop you getting sick, you won't get sick. And if you don't get sick, that has the rather built-in side effect that you're unlikely to die anytime soon. So people are going to live longer, and the idea of somehow thinking that that's a side effect we should apologize for or be scared of is insane.
Yeah, and I'm glad you brought up the longevity escape velocity. Could you dig a little bit more into the fundamental idea behind that and why you chose that as the name for your new institute?
Sure, yes. So the biggest idea, the thing I'm really known for, that I devised about 25 years ago now, was the realization that the most practical and most feasible way to postpone the health problems of late life will be by actually reversing aging rather than by slowing it down. In other words, that essentially if we define aging as the accumulation of various types of damage in the body, molecular and cellular damage, that the body is set up to tolerate only a certain amount of, such that in late life we get sick, then repairing that damage, doing preventative maintenance, is going to be a lot easier than making the body run more cleanly and create damage more slowly than it normally would, which is the focus that the field had prior to my coming along. And the thing about damage repair, rejuvenation, preventative maintenance, is that you buy time. So if you take someone who's already 60, so we say, and you halve the rate at which they're aging, you don't actually benefit them very much. You only delay the onset of age-related conditions by a small amount because they've already got most of the damage that's necessary to give them those conditions. Whereas if you're repairing damage, you're taking them back, then it's going to be a while before they get back to being biologically the age that they were when you treated them. And, of course, you can do this more than once. You can do it as often as you like. It's not quite that simple, though, because the initial first-generation rejuvenation treatments will not be 100% comprehensive. And so there will be certain difficult types of damage that the body is still accumulating, and those on their own, however well you fix the easy damage, the difficult damage is going to make you sick eventually anyway. So the requirement is going to be that scientists will continue, after the development of these first-generation treatments, to develop better ones that chip away at the difficult damage and shift it into the easy category, so that over time more and more of the damage is repairable. And I defined longevity escape velocity as the minimum rate at which scientists are going to have to do that. So basically the whole concept doesn't really kick in until we get our first maybe 20 years of of delay of the chronic conditions of late life, because 20 years is the kind of time you're going to need to give the scientists enough time to make the next big steps. So I believe that 20 years is going to be enough, and once we've got that far, then it's going to be basically vanishingly unlikely that we will ever fall below that rate of progress, because that rate of progress actually slows down over time. It's not like the singularity where you need kind of accelerating change. This is what people have called the Methuselarity, where once you get there, it's just virtually impossible that we would ever fall below that rate again. But it also means that, in contrast to almost everything else in medicine, it's actually going to be easier to reach longevity escape velocity in long-lived species like humans than it is in shorter-lived species. It's going to be touch and go for cats and dogs, and I'm pretty sure that we will never reach longevity escape velocity for mice.
Interesting. And you mentioned the categories of damage. Some may be easier, some probably more difficult with first therapies. Could you walk us through some that you think might be the easiest or were closest to actually addressing that type of damage?
Yeah, so this classification you're talking about, I should probably give a little background. So when I first realized that damage repair was going to be the way to go, it wasn't just that I realized that if you didn't go that way, then you were essentially fighting biology, whereas if you do go that way, you're kind of working in concert with biology. It was also that I drilled down and said, well, okay, how in specifics are we going to do it? And I realized that one very essential first step would be to classify the various types of damage that the body accumulates into a manageable number of categories that would guide the development of damage repair modalities. This is something that was kind of recapitulated in a famous paper that came out in 2013 called The Hallmarks of Aging, which very rapidly became the Bible of the field, and has been cited far more often than any other paper in the whole of the biology of aging this century. But they didn't actually do it very well. They kind of missed the point in a way. They got it right in the fundamental sense that they classified the phenomena of aging and made the whole idea of a divide and conquer approach to aging respectable, which was very helpful to me. But the whole turning that into a coherent project plan, so to speak, for actually bringing the whole thing under control, they didn't really get that right. So what I did was that I said, well, okay, there are these categories. Initially, there were 9. After a couple of years, I was able to reduce it to 7. And for each of these categories, there is a kind of generic approach to doing something about it, to doing the damage repair. So the easiest category back then, which I would say is still the one that's furthest along, is cell loss. Cells dying and not being automatically replaced by the division and differentiation of other cells. And, of course, the natural way to fix that is stem cell therapy. So back then I was already aware that in the 1980s or so, there had been some work in Parkinson's disease in Sweden that seemed very, very promising. So Parkinson's disease, for any of your audience who don't know, it's driven by the loss of a particular type of neuron, dopaminergic neuron, which, as its name suggests, makes this thing called dopamine, which is a rather important neurotransmitter. If these cells die, then we don't make enough dopamine, and you get Parkinson's disease. And they do die much more frequently than most neurons, basically because it's hard work being a dopaminergic neuron. And the treatment, if you put stem cells in, dopaminergic precursor cells, they make new dopaminergic neurons, bang, you don't have Parkinson's disease anymore. And it works incredibly well when it works. In the 1980s, we knew almost nothing about how to manipulate stem cells in the laboratory. And therefore, the way the clinical trial was done was to isolate cells from roughly the right part of the brain of aborted fetuses. And it almost always didn't work at all because they didn't get any dopaminergic precursor cells. But on the rare occasions where it did work, it did indeed work spectacularly well. So now, of course, we know a huge amount about how to manipulate stem cells in the laboratory, and sure enough, the whole idea has been picked up again, and there are something like half a dozen clinical trials around the world going on in that space, and everyone's very optimistic. But of course, the general concept of stem cell therapy can be applied to any organ in which the same kind of thing happens. So, for example, the heart loses pacemaker cells. Now, there we may not need to go this way because the actual non-biological pacemakers work pretty well. But supposing we did, that's another example. The thymus, the thymus atrophies in early adulthood, and that's fine for a long time, but eventually we run out of naive T cells, and that's a bit of a problem. So, you might want to use stem cell therapy to regrow the thymus. Again, we are now looking at other ways that seem to be pretty good at regrowing the thymus, so things may be easier. And that's the general theme that we have seen over the past 25 years since I first put this idea forward, that not only have we made progress in the directions that I described at the time, but also whole new approaches to making to doing the type of rejuvenation of the removal of the type of damage in question have emerged. In the case of cell loss, one important type of cell that's a problem is stem cells of rapidly renewing tissues, like the blood, for example. And my view certainly is that the kind of re-stemification of tissues like that, like the bone marrow, is the main mechanism of action of partial reprogramming, which of course uses the Yamanaka factors to rejuvenate in vivo. So yeah, I mean that's the general principle. So I've spent a lot of time talking about just one of the 7 categories. I could go on about the others, but the general theme is the same.
That's a good segue of many promising therapies are using this partial reprogramming or epigenetic reprogramming. David Sinclair's been a big leader in this, but there's several other companies aiming at this as well. What is your analysis of that technology, and what do you think of their upcoming clinical trials and what to expect and for future uses of this tech?
Yeah, so, I mean, it's still a very young field. We've got to remember that. It's 20 years now since Yamanaka discovered the factors. But of course, in that regard, we were just talking about making genuinely pluripotent cells ex vivo in cell culture, and we've got pretty good at that. We've got better over the years. Just last year, there were a couple of big announcements of how to optimize the Yamanaka factors so that they work 50 times more efficiently than before, and that's pretty good news. But about 10 years ago, a friend of mine from Spain named Manuel Serrano had this crazy idea that maybe it would work in vivo. He did the experiment. He actually just created a transgenic mouse expressing all 4 of the Yamanaka factors. And of course, he knew that if it worked, it would be very bad for the mice because they would have pluripotent cells. But it did work, and they did. They had teratomas all over. Yes. So that was an extremely successful proof of concept. And then a professor in San Diego named Belmonte ran with that and said, well, okay, so let's see what happens if we titrate this, if we just do it a little bit, so as to take the process forward only enough to make the cells a bit more stem-like rather than to turn them into pluripotent cells that don't know how to behave. And it worked pretty well. And that, of course, has led to a whole industry, as you say. There are some really big companies like Altos, which, of course, Belmonte is now the chief science officer at Altos. Serrano is there as well. Various other people involved in epigenetics, like Steve Horvath, are there. So that's all great, and I believe that there's great potential for using partial reprogramming as a tool in rejuvenation. What we do have to remember, though, is that it can't do everything. First of all, there are tissues in which there are just no precursor cells to rejuvenate. So you're not going to be able to do the Parkinson's thing that way because there just are no dopaminergic precursor cells to deal with. then, of course, there are things that don't have to do with stem cells at all, like, stiffening of the extracellular matrix or accumulation of waste products inside non-dividing cells, things like that, that really, you can't expect partial reprogramming to have much of an impact there. But equally, knock-on effects happen. We can never be sure what is going to be essentially potentiated by fixing one type of damage. Most extreme example of that really shook everybody up was about 15 years ago when people started making the first mouse models of senolysis, of removal of senescent cells, which is another of my categories, of course. It turned out to be quite easy to make a model like that. There was a accidentally discovered mutant that allowed mice to make senescent cells a lot faster than normal without accelerating other aspects of their aging, but that was definitely bad for them. They had their lifespan was cut in half. And so one made a model where one could get rid of those cells using suicide gene therapy, basically, introducing a suicide gene into cells that was activated with a simple drug. And it worked. You could get rid of the senescent cells and the mice were happier. But the thing is, they were a lot happier. They were happier in a whole bunch of ways that people didn't think had really anything to do with senescent cells. And this crosstalk between types of damage, I'd always known that it would exist, but that result and other results that have happened since have really shown, I believe, that the magnitude of the crosstalk is much higher than we would have otherwise had any reason to hope. And that's huge. And we're definitely, capitalizing on that in our work at LEV Foundation. We want to make mice live a lot longer with treatments that are initiated in middle age, and we believe that there's a good chance of being able to do so by, we have to hit a lot of targets. We can't just hit one target, but we believe that it's very much within sight, within reach, to hit enough targets that we can really make mice live a hell of a lot longer than we can make them live with, for example, calorie restriction.
Great. And one other big topic is accumulation of DNA damage. And that one, I think there's approaches of limiting it, but towards your approach of reversing the damage that's already acquired, what do you think are the best strategies to approach that one? Because that's unique in every cell.
Yeah, so a lot of people have been very nervous about this one for a long time. They've taken the view that even if we can figure out ways to eliminate, proteinaceous garbage or, cross-links in the extracellular matrix or replenish stem cells or get rid of senescent cells, still, we're going to have this accumulation of noise in the genome at the level of mutations and also at the level of epimutations, of course, the changes to the decorations of the DNA, the methylation and the histones and so on. And, this is going to take cells gradually downhill and we're going to be screwed. So I have always said two things about that. Well, I've said a number of things. Let me start at the top. The first thing I have said since forever is that actually we only need to worry about cancer. And this is because of a phenomenon that I have called protagonistic pleiotropy. So I gave it that name because of the analogy with a very well-known phenomenon in aging called antagonistic pleiotropy, which essentially says that some of the things the body does to itself are bad for you late in life, but they happen because they are continuations of processes that are good for you early in life, and that's why they are selected for, that's why they exist in the genome. And they're not bad enough for you late in life for evolution to have taken the trouble to develop an off switch, right? So we see cases in biology where evolution has taken the trouble to develop an off switch, and we see sometimes where that goes wrong. One of the most high-profile examples of this right now in gerontology is a type of muscular dystrophy called FSHD. The reason it has become high-profile is largely because of one guy named Chip Wilson, who is very wealthy, and because he created Lululemon, And he put a bunch of his money into the HealthSpan X Prize, which I have had a lot to do with over the years. The thing about FSHD is it's exactly this kind of problem. There is a pathway that's involved in muscle development early in life and that needs to be turned off in late life, or else you get muscular dystrophy. And that off switch does exist in all of us, except that, of course, it's genetically encoded, which means that it can be mutated congenitally. And people with FSHD have exactly that. They have a mutation that makes the off switch not work. The stuff that we want to have early in life carries on, and you end up with this disease. So that's fine for most of us, for people who don't have FSHD. But the thing is, the same kind of thing can happen in the rest of biology. Now, it's very unclear that there are any real examples of this in human biology, where there's no off switch and therefore we kill ourselves, so to speak. However, protagonistic pleiotropy is kind of the other way around. Basically, it says that if there are two things that a system does, and one of them kills you a lot younger than the other one, then essentially the other one is going to be protected against unnecessarily well. And the two things in this case are the parts of the genome that have to do with the cell cycle and the parts that don't. So cancer, of course, it starts from one cell, right? It's like one cell gets in a bad way and develops a constellation of mutations that allow it to, ignore cell cycle control mechanisms and keep going and, evade the immune system and break down the extracellular matrix and so on, and you end up, dying of it. Now, we've got lots of defenses, of course, that try to stop that from happening after the mutations have happened, or at least after some of them have happened. But, a lot of it is about stopping the mutations from happening in the first place. And so we have really sophisticated machinery to stop mutations and epimutations from happening frequently enough to make cancers occur. And so all of us are chock full of cells that have a bunch of the mutations necessary to make them cancerous, but only some of those mutations, not all of them, and that's why we don't have cancer. Thing is, though, the same machinery that stops cells from getting cancerous until late in life also protects the rest of the genome, because that machinery doesn't know which bits of the genome are relevant to the cell cycle and which ones are not. And that means that the rest of the genome is protected unnecessarily well. That's a model that I put forward more than 20 years ago, and everything seems to support it. So what this means, of course, is if we can figure out a really, really, really good way to cure cancer, right, which is a big if, but I'll come to that if you like, then we kind of don't need to worry about the rest of the gene, about mutations and epimutations for, a long time, for a lot more than a currently normal lifespan, which is pretty good news. Now, of course, there are caveats here. One caveat is the whole thing that we were speaking about a moment ago, epigenetic noise and loss of identity. But as I said, I think that a large part of I think most of the reason why epigenetic reprogramming is effective as a rejuvenating modality is the restoration of stemness in certain tissues like the bone marrow. And there, we can say that, the mutations and epimutations are not causing a disease per se, they are essentially causing cells to die or differentiate. One could say the same thing about senescent cells. Maybe senescent cells are, at least in large part, created as a result of DNA damage, and there's not very many of them, and the immune system gets rid of most of them, but not all, and so on. And so we like to get rid of these things, but cancer's the main problem. I don't think we need an oracle. I don't think we need to have, clever nanobots that go into cells and identify deviations from the correct genetic and epigenetic sequence and fix them because the nanobots know what the correct sequence ought to be, we can just stick with curing cancer and we'll be fine.
Great summary. Are you familiar with MatterBio Sciences and their approach to DNA damage? What do you think about their approach? I know it originally started with something like that nanobot that would ultimately correct everything, but that's an engineering nightmare and difficult to implement, but seems their approach now is more slowing down the accumulation or empowering cells to boost up that system you mentioned that's already in place, especially early in life.
Well, so there's a lot going on in this space right now. So MatterBio is a very interesting company because it really has two directions, which is something that is normally frowned upon in the private sector, but it came together because of a husband and wife team. Both of them are professors at Einstein, and one of them was focused on DNA damage and repair, and the other one was focused on cancers and the use of bacteria to target cancers. And the two technologies have come together in this one company. So, yes, it's a very exciting company. Well, honestly, I would say that the most exciting thing in this area is really not what MatterBio are doing. The most exciting area, it comes out of Germany with the discovery of this extraordinary thing called the dream complex, which is a protein complex that does not protect the DNA. It does the opposite. It actually inhibits the activity of DNA repair of various types. And it is active in pretty much all of our cells. The one place where it's definitely not active is the germline. And of course the germline needs to have a considerably higher degree of fidelity of DNA maintenance than the soma because it has to deliver pristine DNA to the offspring. So we can look at the dream complex as potentially an example of this antagonistic pleiotropy that I was mentioning a moment ago in relation to cancer. We still don't really know, but it looks like it. So first of all, we have to ask, what's good about suppressing DNA repair even early in life? And the best answer we have so far is that really early in life, during embryonic and fetal development, you have to do cell division really, really fast. There's a lot that has to go on. In some organisms, like fruit flies, for example, you have to do it so fast that they give up on the idea of even having individual cells until, for a period during embryonic development. So the whole embryo is a kind of syncytium, where the DNA is dividing faster and faster, but most of the rest of cell division is kind of dispensed with. We don't do that. But still, it has to be really fast. And the thing about DNA repair is it takes time. DNA repair enzyme has to find the lesions to fix and do the fixing, and, it slows things down. So the theory is that DREAM exists in order to allow cell division to be really fast early in life, and that then, DNA repair is still good enough throughout the rest of life that evolution has not developed the off switch that we're talking about here to make DNA repair better. And so people are pretty interested in the idea that we might be able to fix that. What nobody's saying, however, and which I don't think we can say, is that if we were to fix this by suppressing the activity of DREAM, then we would automatically live a lot longer. Because there are too many things that go wrong that, even taking into account the knock-on effects that I was mentioning earlier, just will not be appreciably slowed down by having better DNA repair.
Excellent. And to follow up, Chris Bradley of MatterBio was one of our previous guests, if you want to check out his podcast before. Mm-hmm. Great conversations. And to sum up DNA damage, do you think there are other major forms that we do not yet know about or just have difficulty classifying? Or do you think we have a pretty good roadmap and grip of the major categories of damage that are causing aging?
I would say that falls into two very contrasting categories. I think we certainly have a very thorough understanding of the ways in which the cell limits the accumulation of mutations. I mean, dream was only discovered recently as an influence on the activity of those things like base excision repair and non-homologous end joining and all these various things. But we know an awful lot about those things. So that's pretty good, and I'd be pretty surprised if we discover new types of DNA lesion that were not previously known or anything like that. However, the contrasting one is the epigenome. Epimutations are very ubiquitous. They happen all over the place. Changes to the methylation of CpG dinucleotides, changes to histone modifications, changes to higher level chromatin structure. And as I mentioned, these things do have an impact on cellular function. Thing is, we know basically nothing about how the cell controls that. For the longest time, we didn't even know how, if at all, cells remove methyl groups from CpG dinucleotides, the demethylation thing. Eventually people discovered this thing called hydroxymethylcytosine that is an intermediate, and that led to an understanding of some of the things that are going on. But it's painful. And even de novo methylation, as opposed to maintenance methylation, where you take, you put a methyl on the cytosine that's opposite the guanine that already has the cytosine methylated. Even that took a while to be discovered. So at this point, we are absolutely at square one in understanding what's going on. But I think it's fair to say that something's going on, that we are seeing epigenetic noise accumulate at a slower rate than it would if the cell were not trying to slow it down. We just don't know how it's trying to slow it down. But a good evidence for the fact that it is slowing it down is the amount of epigenetic noise that we see. And this is actually something I'd like to talk a little bit more about, because it brings us to the topic of epigenetic clocks. So, of course, it's very fashionable now, and rightly so, to use the epigenome as a way to measure biological age. And there's some way to go to make this thing really work. People have developed clocks that are only for one organ. People have developed clocks that seem to work across the whole of mammals. There's lots of different ways to do it, and they've all got promise. But, of course, the goal of these clocks is to determine whether therapies are working, to have a kind of surrogate endpoint for an initial study on some new drug, for example. And, it's not really looking all that promising, I would say, that we can create clocks that we can really rely on in this regard. Because, of course, these clocks are trained on data sets that do not include the thing that you haven't developed yet. Right? So there's a new type of clock that's just started to be talked about and explored. Clocks that have happened over the past decade have all been about CpGs in which the amount of methylation changes with age, ideally at a fairly linear rate. And so you look at, variations in that rate. But another way of doing it would be to look at CpGs that don't change on average during aging at all, in particular at ones where, there's probably a good reason why they don't change, because they're in promoter regions of genes, things like that. And then to look at the amount of variability in their methylation across a tissue, across a variety of cells from a given type. So what you're doing there is you're measuring how well the body is controlling the methylation of the particular site that you're looking at, the particular locus you're looking at. And sure enough, it turns out there's more variability later in life. As aging happens and stress increases, and the cell is generally worse at doing shit. So I believe that that's got enormous potential, that it's a much more global, much more intrinsic measure of cellular health than the kind of clocks that we've got at the moment, and therefore that it's got a much better chance of being a reliable surrogate endpoint.
Excellent. And you mentioned this dysregulation that happens as you age, natural disorder comes in. It's been observed that the transposable elements activate both in their expression and their transposition. Companies like Rome have some approaches towards this. What are your thoughts on that? One, global transcriptional dysregulation, but more specifically with transposable elements.
Yeah, so this is an exciting idea. It's been explored. We were involved, my previous foundation was involved in funding a little bit of work in Andrei Gudkov's lab on this. We are I'm definitely paying attention to this whole area. So the basic problem here is that there's a lot of this stuff in the genome, these retrotransposons that have kind of invaded the genome and have been hard to get rid of during evolution. And so rather than getting rid of them, evolution has found ways to suppress their activity, so they just sit there and don't do anything. As with everything else in the epigenome, that the quality of that suppression diminishes over time, so that here and there the occasional retrotransposon starts to be transcribed. And once it's been transcribed into RNA, of course, it can get reverse transcribed into, or it reverse transcribes itself into DNA somewhere else in the genome, and suddenly you've gone from having what looked like a perfectly harmless epimutation in something that wasn't a gene, you've got mutations in perhaps important genes. So this is definitely a process. But again, everything I said about protagonistic pleiotropy, about the essentially cancer being the only thing that matters, applies just as much to retrotransposon-mediated mutations as it does to any other type of mutation. So again, I'm not terribly worried. Except insofar as we do actually have to, as such, cure cancer. So we can come to that if you like.
Mm-hmm. Well, do you want to go into that? You mentioned the cure for cancer, and it was a big if in your mind, but what do you think are the timelines or top approaches to actually do that? Because we've made many progresses towards specific types of cancers, but to broadly stop all cancers, what are your thoughts on that?
Yeah, so when I started thinking about all of this 20 more years ago, I didn't like the way it was being talked about. It seemed like people had said, well, okay, you shouldn't think about cancer as a disease. Cancer is this family of diseases that, each have their own characteristics. And of course that's true. We see in different cancers, we see certain kind of Achilles heels, certain genes in which mutations cause a particular type of cancer. That's certainly true. But also, because of the inherent genomic instability that allows cancers to get where they are, if you try to focus on those Achilles heels, cancers are just going to find a way to escape, to get out and pretend to be a different kind of cancer or whatever. So I felt, it's kind of missing the point. And what we should be doing is looking for ways, for things that cancers have in common, that are as universal as possible across all cancers. In other words, essentially going back to the idea of thinking of cancers as, for practical purposes, just one disease. And I realized pretty quickly that there was really only one game in town in that regard, which was the telomere. That every cancer has no choice but to figure out a way to maintain telomeres, or else it will divide itself into oblivion way before it gets big enough to kill you. And of course, this has been supported by a bunch of cross-species work in which it has been very clearly shown that large mammals have this trick of essentially which I've called telomere thrift, in which you essentially express telomerase, the enzyme that maintains telomeres in the germline. You express it in somatic cells at the absolute minimum amount that you can get away with, so as to maximize the amount of epimutation in the promoter regions that would be required in order to turn it on sufficiently to make a cancer. And this works pretty well, as far as we can tell, in large mammals. In small mammals, it doesn't work at all, because small mammals can be killed by small cancers, which don't have to divide very much and therefore don't get much telomere shortening, even if the telomere is not being extended by telomerase. And so sure enough, mice they just say, well, fuck it, let's just express a bunch of telomerase anyway because it's an easier way to maintain stem cells and so on.
Mm-hmm.
Whereas humans do not do that. So, what does this mean? Well, can we control the telomere? I realized pretty quickly that if we try to do it specifically in cancer cells, we're going to be screwed. It's not going to work because cancer cells are just very ingenious. They're very good at figuring out how to masquerade as non-cancer cells and to sidestep whatever selection criterion we are choosing to have. So I said, right, we've just got to kill off telomerase completely in all cells. And that's going to have a side effect or two. It's going to cause the stem cells in our rapidly renewing tissues, like the gut lining and the blood and the epidermis and the lining of the lung. It's going to cause these things to atrophy, but not for a while. Probably not for a decade or so, because the stem cells, the long-term repopulating stem cells in these tissues, only divide once every few months, and so that's a long time before they're going to get in trouble. And so I thought, well, okay, we can just live with this because we can do stem cell therapy. It won't be easy. And indeed, various people who were pretty positive about everything else I was saying to do with damage repair, they said, listen, no, this is just never going to work. Mm. And honestly, I always accepted that it was a really ambitious approach. But I said, well, better to be ambitious than to give up, right? So that's the approach that I decided we should pursue. Now, there is one problem here that I haven't yet mentioned, which is ALT. So about 10% of human cancers do not express telomerase, this enzyme that is expressed in the germline to maintain telomeres. Instead, they have this extraordinary mechanism that was discovered at least 30 years ago now and is still not really very well understood. ALT stands for alternative lengthening of telomeres, which tells you how little we understand about it. So we've got to fix that as well. But it might not be terribly hard to fix. ALT seems to rely on an awful lot of machinery that needs to be just right. And so it may not be too hard to do that. But we'll come back to that. In the specific case of telomerase, though, which does account for 90% of human cancers, there was an enormous breakthrough about a decade ago in Texas, in the lab of a fantastic researcher in telomeres named Jerry Shea. He found a molecule which is a nucleoside analog, something that gets incorporated into DNA. And it's got a sulfur atom in it. It's called 6-thiodeoxyguanosine. And it's incorporated into the DNA, and it messes it up. But here's the thing. It's only incorporated into the DNA by telomerase. DNA-dependent DNA polymerases recognize it as something foreign, something unlike the real thing, and they have nothing to do with it. They do not incorporate it. And that applies to DNA replication, it applies to DNA repair. The only enzyme that incorporates this thing into the DNA is telomerase. So that means that you can turn telomerase into a suicide gene. You can give this molecule, and cells that are expressing high levels of telomerase will rapidly incorporate it into the DNA of the telomere, and the telomere will no longer work. In particular, the telomere will no longer form what's called a T-loop, which is a structure at the end of the telomere that allows the telomere to do its job. The job of a telomere, which is actually somewhat misunderstood by the general public, is not just to be information-free DNA as a kind of buffer to stop DNA replication from destroying genes. What it is it's a structure that stops the cell from misinterpreting the end of a chromosome as a broken chromosome. If the cell gets that wrong, then it joins chromosomes together end to end. And chromosomes that are joined together end to end are very bad for the cell. The cell dies very quickly. So basically that's what this drug does. It causes the telomere to be unable to form this loop, and chromosomes do get joined together end to end, and the cell is history pretty quickly. So this is enormously preferable. to the original concept that I put forward, because it kills cells quickly. It means that you don't need to wait for the cell that's no longer got its telomerase to, divide itself into oblivion. You can just kill the thing off quickly. Now, furthermore, we have to remember that cancers that rely on telomerase make a lot of it. And they make vastly more of it than any of our stem cells do. Even the stem cells in the gut or the blood, the bone marrow, they make trace amounts. It took until the 1990s for anyone to be able to even directly measure how much telomerase was being made because it was so small. So basically that means there's a really good therapeutic window. You can really knock your cancers on the head before doing any significant damage to your hematopoietic stem cells, for example. And so this has moved on slowly but surely, and it's now in clinical trials in Australia. The people who are pursuing it are actually combining it with cancer immunotherapy to make it even more effective, but I'm very optimistic.
Excellent. I'll follow that research and look into it a bit more. So one thing, just to segue...
The company in question, just to emphasize, I don't have any financial involvement with it. The company is called Maya, M-A-I-A.
M-A-I, excellent. Illusion in Sanskrit. So for one thing I like to ask all of my guests is how, as if it has, science fiction influenced your pursuit of longevity or aging and science? And if it has, could you give a recommendation to the audience?
Yeah, it really hasn't really influenced mine. What's quite surprising is it didn't influence my wife either, because she was a big science fiction fan. She would read, Heinlein and Asimov and so on all the time. And, they wrote quite, interesting things about a post-aging world, including, making very positive things. The main attitude that I have to science fiction in relation to aging is negative. I think that it makes the science fiction in the current world, in the past few decades, that has focused on a post-aging world has made my life a lot harder, because it's almost always dystopic. It's always like they create some kind of storyline that says, okay, we've created a post-aging world, it's a terrible place to be for this or that reason, we have to kill everybody at the age of 30, or, it's only available to wealthy people. And so, it makes my life harder because it entrenches the inherent, what I've called the pro-aging trance. This attitude that you need to convince yourself that aging is a blessing in disguise in some way in order to put it out of your mind and get on with your miserably short life and not be preoccupied by this terrible thing that's going to happen to you. And people want to do that because they're terrified of getting their hopes up, and listening strongly to people like me that say that, we might actually be able to fix aging in time for you, which is it used to be a sensible thing to do, to put it out of your minds, however irrational your rationalization might be, because, we didn't have a plan. But since I came along, we do have a plan, and therefore this is really slowing us down, and I really don't like that. So there's a really important need for blockbuster movies and so on that portray aging in a positive light, portray the end of aging in a positive light, and that hasn't happened yet.
I agree. There's definitely a huge trend from Heinlein and even Star Trek, for example, that have this positive future versus today really covered in dystopia. So I also don't like that trend, but still am inspired by a lot of the older works and some newer works. You mentioned Robert Heinlein. Have you read Time Enough for Love?
Oh, of course, yes.
Yeah, the Lazarus Long series, and a very interesting aspect of the Howard Foundation and naturally breeding long-lived families, and it's a great story that implements the treatment as well. I think that's a great novel I'd recommend for people to read interested in aging.
Yeah, the first one that I read that my wife actually gave me was a novel called Trouble with Lichen, which was all about how someone discovered that there was a type of lichen that had a really effective anti-aging chemical in it, and they recognized that they couldn't market it as an anti-aging drug because of the pro-aging trance, basically, so they marketed it as a cosmetic, and that worked very well.
So speaking of that, of supplements or cosmetics, do you personally implement any supplementation or vitamins or lifestyle practices or diet? Could you comment on what you're implementing short of these therapeutics that are coming along the way?
Yeah, so I'm one of those repulsively lucky people. I've always been able to eat and drink exactly what I like and nothing happens. And I have also, because of being a high-profile guy, I've been able to get access to very high-end measurements of my biological age for the past 20 or more years. And I always come out ridiculously younger than I really am. So I've always taken the view that the right thing for me to do is to be rather conservative. And, if it ain't broke, don't fix it. And not to try to do any better. I do not recommend that to the average person, of course. I think that everyone ought to find out as much as they can about their biology and to act on what they find. And actually, just as of not very long ago, I have started taking one supplement that's important, a type of vitamin B12 called methylcobalamin. And the reason I take it is because I got my genome sequenced. And it was established that I have a variant of a gene called MTHFR that it predisposes to having high levels of something in the blood called homocysteine, which is generally considered to be not a good thing. Now, I've known for a long time that I have high homocysteine. I don't have any of the things that tend to go with that, so I seem to have a good way to compensate. But nevertheless, I'd rather not have the high homocysteine in the first place, so I am taking this one thing that is well known to work quite well to reduce homocysteine.
Excellent. Anything else you learned from your genome, from having that sequence that you've put into practice?
No, everything else was basically normal. But of course, the thing is that there's constantly new information about, which genetic sequences predispose to which kinds of age-related problems. So, that keeps getting updated.
Excellent. Just for some closing remarks towards the end of the show, what do you think most people asked a question about that they don't? Speaking of this dystopia or improper mindset that a lot of people have, or perhaps flawed mindset from pop culture or media, what do you wish you could ask them or talk to them about?
Well, I don't have to wish because I spend my time talking to people. I guess I would say that the single biggest thing that matters to me and seems not to matter to most people is other people. I describe myself as a humanitarian first and foremost. That, the thing that gets me out of bed in the morning is not thinking, the more I work on aging, the better my own chances of, making the cut and, benefiting from the achievement of longevity escape velocity. Because I recognize that even though I am doing quite a lot, to hasten the defeat of aging, nevertheless, in numerical terms, the amount by which I am improving my own chances is pretty modest. A few percent if I'm even quite optimistic. Because, it's a few years I might bring forward the defeat of aging, but, I might already be young enough that I'm going to make it anyway, even if I were doing something else with my life. Or conversely, I might be too old, so that however fast it happens, that won't be in time for me. So that's a difficult thing to get excited about. Whereas, if I look at the whole world, 110,000 people die of aging every single day. So every day that I bring forward the defeat of aging, I'm saving 30 World Trade Centers, and that's quite easy to get out of bed for. So I like to try to impress on people that the reason they should think of the war on aging as important is because of humanity in general. Not because of themselves, not even because of their loved ones, who are relatively few in number, but because of people in general.
And having that mission may be why you come in at a lower biological age when you assess that. I really believe that having that mission in life keeps you going.
Having purpose is definitely good for stress levels, and having low stress is good for one's aging. Sure.
Yeah. So to close, if you have one message for the next generation of scientists interested in aging, what advice would you give them?
Well, That's actually a really difficult one because there's so many different things to do. If we're talking about when I advise young scientists on which directions to go, for example, if they're at high school and they want to know what kind of degree to do, I always say do a general biology degree rather than going into genetics or biochemistry or whatever, because aging affects the body at every level of organization. In order to understand aging well enough to be able to do something about it or contribute to the effort to do something about it, you've really got to understand biology throughout. So you've got to have a good grounding in biochemistry and cell biology and genetics and pharmacology and physiology and anatomy and everything to do a general biology degree. After that, of course, if one's going into research, doing a PhD and so on, then, a lot of the issue is psychological. One needs to have a very thick skin and a lot of determination to get through a PhD because everybody has long periods of frustration where things aren't working. And so one needs to focus on things that one is really, excited by, really, really influenced by. And also, I often say, work in a relatively small lab because otherwise you will be a small cog in a large machine and you won't have much autonomy. That, of course, isn't always true. I think George Church is actually a great example of someone who has given the younger members of his flock a lot of autonomy and freedom to get stuff done that's worked out very well. But I think he's the exception in regard to people with large labs. So that's what I would say.
Excellent. Well, great conversation today. Really appreciate having you on. Thanks for joining.
It's my pleasure. Thank you for having me.